Hello students, welcome to Edupedia World Videos. My topic for the presentation is fourth section of the chapter Transport and Plans. In this section of the video, we will discuss about the means of transport such as diffusion, facilitated diffusion, passive symport, antiports, active transport, water potential, guttation, osmosis, plasmolysis and deplasmolysis. So first we'll start with diffusion. Diffusion, students, it is a movement by diffusion is passive and maybe from one part of a cell to another or from one cell to another cell over short distances, say from intercellular space of the leaf to the outside, and no energy expenditure takes place. See, when I say that movement by diffusion is passive, then that means no energy expenditure will take place. In diffusion, molecules they move in random fashion. See, here in these are the molecules present on the high concentration, so they move in a random fashion, and the net result being substances moving from region of a higher concentration to the region of lower concentration. Okay, and please note that diffusion is a slow process and is not dependent on the living system, and diffusion is obvious in gases and liquids, but diffusion in solids rather than of solid is more likely. And diffusion is very important to plants since it is the only means for gaseous movement within the plant body. Okay, so diffusion, you can say that it is a movement of a molecule of gases, liquid, solutes from region of higher concentration to the region of lower concentration, which is known as diffusion. As you can see that this is high concentration solution, so and these are the solutes which passes from high concentration solution to the low concentration area, and this is the net movement of molecule or ion of a given substance from region of higher concentration to lower by virtue of their kinetic energy. That means these are the solutes that move in a random manner by virtue of their kinetic energy. So it is the movement of molecules from high diffusion pressure to low diffusion pressure. That means this is high diffusion pressure area and this is low diffusion pressure area. So solutes will move from high diffusion pressure area to solution or to the area of low diffusion pressure. Okay, see, this is passive transport because it does not require expenditure of energy. That's why it is called as passive, because it occurs in a random fashion by virtue of their kinetic energy. Okay, see, this is diffusion, which is showing that from a high concentration area, solutes are moving from high concentration area to a region of low concentration, and this is the protein which is embedded in the plasma membrane. See, this is plasma membrane. Okay, and this is a protein which is embedded in the plasma membrane, and which is facilitating the transport of solute from this region to here to this region. So that's why it is called as facilitated diffusion. Okay, now we'll talk about the facilitated diffusion, students. Facilitated diffusion is a type of a passive transport that allows substances to cross membrane with the assistance of special transport proteins. See, these are the transport proteins which are embedded in a plasma membrane, and facilitated diffusion is a type of a passive transport that allows substances to cross membrane with assistance of special transport protein. Some molecules and ions such as glucose, sodium ions, chloride ions, they are unable to pass through the phospholipid bilayer of cell membrane. See, cell membrane or plasma membrane, it is made up of phospholipid bilayer. So it is seen that some molecules and ions such as glucose, sodium ion, that is Na+, chloride ion, that is Cl- ion, they are unable to pass through the phospholipid bilayer of a cell membrane. So through the use of ion channel protein and carrier protein, see, this is ion channel protein, or it is also called as carrier protein, which are embedded in the cell membrane, these substances can be transported into the cell. An ion channel protein, it allows specific ions to pass through the protein channel. Okay, And the ion channels, they are regulated by the cell, and they are either open or closed to control the passage of a substance into the cell. Okay, see, this is the outside cell and this is inside cell. See, this is low concentration area of a solute and this is high concentration area of solute. So from inside cell of the cell, which is high concentration, it solutes, it starts to move outside of the cell, which is low concentration region, with the help of these carrier proteins or channel proteins. So the ion channels, they are regulated by the cells and they are either open or closed to control the passage of a substance into the cell. And carrier protein, it bind to the specific molecule, it changed the shape. See, it is when it takes up the solute, it changes the shape. Okay, and then it deposit the molecule across the membrane, then it just ejects out the solute to the outside part of a membrane. Once the transaction is complete, the protein, it returns to their original position because it has changed its shape to take up the solute so that it can transport to the outside of a cell membrane. Then it reaches or it returns to its original position once the transaction is complete. Okay, as you can see that this is facilitated diffusion and it is passive transport. That means no energy expenditure takes place while transporting solutes from higher region to the lower concentration region. Okay, now we'll talk about the Symports and Antiports, students. Okay, some carrier or transport proteins allow diffusion only if two types of molecules move together. In Symport, both molecules cross the membrane in the same direction. See, these are the two molecules A and B which are crossing the membrane in only in the one direction. So Symport, it transports the solute and a co-transported solute at the same time in the same direction. And this is Antiport, wherein this is one solute and this is another solute which is present on the either side of the membrane and they move in the opposite direction. So in an antiport, they move in an opposite direction. Okay, and when a molecule move across a membrane independent of another molecule, then that process is called as uniport. So I hope all these three are clear to you. These are the transport molecules: uniport, symport, and antiport. So some carrier protein, it allow diffusion only if two types of molecule move together. When both the molecule cross the membrane in the same direction, then it is called as symport. And when the molecule move in the opposite direction, then it is called as antiport. And when a molecule move across a membrane independent of other molecule, then the movement is called as uniport. So uniport, it transports one solute at a time. Symport, it transports solute and co-transported solute at the same time in the same direction. As you can see, A and B are moving in the same direction. And antiport, it transports the solute in or out, or co-transported solute in the opposite direction. One goes in the other, goes or out, or vice versa. Okay, now we'll talk about the factor that influences rate of diffusion, students. See, diffusion rate are affected by gradient of concentration, permeability of membrane, separating them, temperature, pressure, and density of diffusing substances. Okay, so first we'll discuss about the temperature. Obviously, when you increase the temperature, it leads to the increase in the rate of diffusion. Then we'll talk about the pressure. The rate of diffusion of gas is directly proportional to the pressure. So the rate of diffusion, it increases with the increase of pressure. That means when you increase the pressure, obviously rate of diffusion will increase. So that means it is in the direct proportion with each other. Then we'll talk about the third factor, and that is size and mass of diffusing substance. Diffusion of solid is inversely proportional to the size and the mass of a molecule and ion. That means if the size or the mass of a molecule is larger, then diffusion will be slower. And on the contrary, if the size and the mass of a molecule or ion is smaller, then diffusion will be rapid. Okay, so they are in the inverse proportion with each other. Now, the fourth factor is density of diffusing substance. Larger the molecule, slower will be the rate of diffusion. According to the density of the diffusion of a substance takes place in the following manner. That means gases will diffuse faster, then liquids, and then solid. That means solid are the larger and the compact molecule, and hence rate of diffusion will be slower. But on the other hand, gases can be diffused rapidly or faster. And the fifth factor that influences rate of diffusion is density of the medium. The rate of a diffusion is lower if the medium is concentrated. That means if the medium is highly concentrated, then the rate of diffusion will be slower. Thus, a gas would diffuse more rapidly in vacuum than in air. I repeat, a gas would diffuse more rapidly in vacuum than in air. Substances in solution also diffuse, but at a much slower rate than gases. And substances in solution diffuse more rapidly from region in which their concentration is higher into region of lower concentration. Now let's say if two solution of sugar of different concentration are in contact, sugar molecule diffuse from higher to lower concentration of solute, and water molecule will diffuse from higher to the lower concentration of water until equilibrium is established or attained, when two solutions become of equal concentration. Okay, now we'll talk about another factor that influences rate of diffusion, and that is DPD, that is diffusion pressure gradient. I repeat, DPG, that is diffusion pressure gradient. The rate of diffusion is directly proportional to the difference of a diffusion pressure at two ends of a system, and it is inversely proportional to the distance between two. Okay, so it is the rate of diffusion, it is directly proportional to the difference of a diffusion pressure at the two ends of a system, and it is inversely proportional to the distance between the two. Okay, now students, what's the significance of diffusion? Why it takes place? So gaseous exchange during the process of photosynthesis and respiration takes place with the help of diffusion. The process of diffusion is involved in the transpiration of water vapors. See, this is highly concentrated solution and this is low concentrated solution, and this green dots are representing solute. So solute will move from high concentration solution to low concentrated region. Okay, so the process of diffusion is involved in the transpiration of water vapors. Aroma of flower is due to diffusion of volatile aromatic compound so that it can attract pollinating animals such as bees. Okay, so that's the significance of diffusion. And during passive salt uptake, the ions are absorbed by process of diffusion. Diffusion, it helps in the translocation of food materials. Gaseous exchange in submerged hydrophyte is taken place by general surface of a cell through diffusion. Okay, so these are the significance of diffusion. Now we'll talk about the imbibition. See, it is a special type of diffusion. Okay, it is nothing, it is just a diffusion, but it is a special type of diffusion which includes the absorption of water by solid such as or you can say called colloids, resulting in enormous increase in volume. So imbibition is a special type of diffusion when water is absorbed by solid, that is colloid, cause them to enormously increase in volume. The classical example of imbibition are absorption of water by seed. See, when you immerse seed in water, then it imbibes. Okay, or you can see when you place dry wood inside water, then it shows imbibition. So the pressure that is produced by swelling of wood had been used by prehistoric man to split rocks and boulders. If it were not for the pressure due to imbibition, seedlings would not have been able to emerge out of the soil in the open. They probably would not have been able to establish. Okay, so imbibition is also a diffusion since water movement is along a concentration gradient. Okay, see, this is the beaker wherein water is there, and you put dry seeds, and after some time you see the seeds have become swollen, and that's the because of the imbibition. Root and shoot emerges from the germinating seed. Okay, and please note that imbibition is also diffusion, since water movement is along a concentration gradient. Seed and other such material have almost no water, hence they absorb water easily. And water potential gradient between absorbent and liquid imbibed is essential for imbibition. In addition, for any substances to imbibe any liquid, affinity between absorbent and liquid is also a prerequisite. Okay, now we'll talk about the diffusion pressure deficit, that is DPD, and suction pressure. Suction pressure and DPD, that is diffusion pressure deficit, they are same terms. So don't get confused with it. Okay. So the term diffusion pressure and diffusion pressure deficit were put forth by B.S. Mayer. He was scientist in year 1938. Originally, DPD, that is diffusion pressure deficit, was described by term suction force. By whom? By Swakrafft. He was scientist. Or suction pressure, that is SP. Renner coined it. Okay. So DPD is nothing, it is a suction pressure. Nowadays term water potential is used, and by which sign we represent water potential? By Y. Okay, so water potential, diffusion pressure deficit, and suction pressure, they all are same, so don't get confused with it. Okay, which is used, which is equal to DPD but negative in value. That means water potential is always in the negative value. Water potential is a tendency of water to move from one place to another. See, this is the membrane. Okay, this is the water molecule, this is the solute molecule, this is low water potential area, and this is high water potential area, but this is low concentration of solute. That means solute are very little. See, these dots are representing solutes, and these are water molecules. So net movement of water takes place from high water potential to the low water potential. Okay, diffusion pressure deficit or suction pressure. Each liquid has a specific diffusion pressure. That means every liquid has its own diffusion pressure. Pure water or pure solvent has maximum diffusion pressure. Please note that a pure solvent or a pure water, it has maximum diffusion pressure. The amount by which diffusion pressure of water or solvent in a solution is lower than that of pure water or solvent, then it is called as DPD. DPD is what? It is amount by which diffusion pressure of water or solvent in a solution is lower than that of a pure water or solvent. So it is called as DPD. Because of the presence of diffusion pressure deficit, that is DPD, a solution will always tend to make up the deficit by absorbing water. Hence, diffusion pressure deficit is water absorbing capacity of a solution. I repeat, diffusion pressure deficit, that is DPD, is the water absorbing capacity of a solution. Therefore, DPD can also be called as suction pressure. See, this is the relation between diffusion pressure deficit and the entrance of a water in the soil. See, DPD is higher in this cell and DPD is lower in this cell. So obviously from this area to this area, water will move from here to here because it has more DPD, that is diffusion pressure deficit, and that's why it is called as suction pressure. So diffusion pressure deficit is a water absorbing capacity of a solution. So when it is 4, that means it has more water absorbing capacity than DPD 2. Okay, that is DPD equals to 2. So this has little less water absorbing capacity of this solution. Okay, now we'll talk about the permeability of a lipid bilayer. See, students, permeability is a degree of diffusion of gases, liquids, and dissolved substances through a membrane. Different type of membrane may be differentially permeable to different substances. Normally, permeability of a given membrane it remains unchanged, but it may change with alteration in the environmental condition of the cell. So as you can see that plasma membrane, it is made up of lipid bilayer. So these two layers are showing lipid bilayer of a plasma membrane. These are the hydrophobic molecule that can easily go from this place to here. So that shows that this lipid bilayer is more permeable to oxygen, carbon dioxide, N2 and benzene, whereas a small uncharged polar molecule such as H2O, urea and glycerol, it cannot move inside, or you can say a little part of it can move inside, and the large uncharged polar molecules such as glucose and sucrose cannot, or it diffuse across membrane. And ions such as H plus, Na plus, H2CO3 minus, K plus, Ca2 plus, Cl minus and Mg2 plus, it cannot move inside the plasma membrane, and thus it bounces back from it. So this is about the permeability of a lipid bilayer of a plasma membrane, wherein a smaller and more hydrophobic molecule it diffuse across membrane more rapidly. Okay. Now we'll talk about the type of a membrane on the basis of their permeability. First is permeable membrane, wherein it allows free passage of solvent, that is water, and most of the dissolved substances. Example is cell wall in plant cells, and filter paper is made of pure cellulose. It also functions as permeable membrane. So this is a plant cell, this is the cell wall, which allows free passage of solvent and most of the dissolved substances. Okay, so cell wall is an example of permeable membrane, and it allows free passage of solvent and most of the dissolved substances. Okay, now second is impermeable membrane. Impermeable membrane, wherein they just don't allow the entry of water and dissolved substances and gases inside it. An example is suberized wall of cork cells, cuticle layer of leaf. See, cuticle is always present outside the leaf, so it does not allow anything to go inside it. So it is an example of impermeable membrane, wherein they don't allow the entry of water, dissolved substances and gases. Okay, then comes semi-permeable membrane. These membrane it permit the movement of solute or solvent molecules, sorry, only through them, but it prevents the movement of solute particles. And examples are egg membrane. See, this is the egg membrane. Okay, animal plasma membrane is also an example of semi-permeable membrane, wherein they allow movement of solvent molecule only through them, but it prevent the movement of solute particle. That means it does not allow solute particle to move inside it, but it allows or it permits the movement of solvent molecule inside of it. Okay, so this is semi-permeable membrane. Now we'll talk about the selectively or differential permeable membrane. Okay, this type of membrane it allows selective passage of solute along with solvent through them. For example, osmotic diffusion of water through selectively permeable membrane. It starts from high water potential to low water potential. And many biological membranes such as cell membrane, tonoplast and membranes surrounding the subcellular organelles, they are called as selectively permeable. And a non-living selectively permeable membrane is cellophone. So this is selectively or differentially permeable membrane, wherein it allows selective passage of solute along with the solvent through them. I repeat, in selective or differential permeable membrane, it allows selective passage of solute along with the solvent through them. Okay. Examples are many biological membranes such as cell membrane, that is plasma lemma, tonoplast. Tonoplast is a membrane of a vacuole, and membrane that surrounds the subcellular organelles, they are selectively permeable. Okay. See, this is the outside part of a plasma membrane. This is the lipid bilayer of a plasma membrane. This is the inside view. So some molecule can cross the membrane while others cannot. So this is selective permeability. Okay, and this is the fourth type of a membrane on the basis of their permeability. Now we'll talk about the osmosis, students. Osmosis, it was discovered by App Knowledge, and it was and he also coined the term osmosis. Okay, so the plant cell is surrounded by a cell membrane and a cell wall. We very well know that, and the cell wall is freely permeable to water and substances in solution, hence is not a barrier to movement. In plant, the cell, it usually contain large central vacuole, right, whose content or vacuolar sap it contribute to the solute potential of the cell. And in plant cell, cell membrane and the membrane of a vacuole tonoplast together are the important determinants of a movement of a molecule in or out of the cell. So osmosis is the term that is used to refer specifically to the diffusion of water across a differentially or semi-permeable membrane. And osmosis means a pushing or impulse. First of all, Traube in year 1867, he used copper ferrocyanide, and he developed a semi-permeable membrane to show its utility in the osmosis of plant physiology. And first time, Pfeffer in 1887, he developed the osmoscope by using semi-permeable membrane. Osmosis is a special type of a diffusion of a liquid when solvent moves through semi-permeable membrane from a place of higher diffusion pressure to a place of lower diffusion pressure. So as you can see that it is low sugar concentration. This green color circles are showing sugar, and these blue dots are water. So this is low sugar concentration and high water concentration solution, and this is high concentration sugar concentration area, and this is low water concentration area. So from here to here osmosis is taking place, and this is semi permeable membrane or selectively permeable membrane. So it is the migration of a solvent from a hypotonic solution, that is of lower concentration, to hypertonic solution which is of higher concentration through semi-permeable membrane to keep the concentration equal. So this is before osmosis, wherein no exchange is taking place, and this is after osmosis, and this is the semi-permeable membrane. And from you can say that it is a migration of solvent from hypotonic solution to the hypertonic solution through semi-permeable membrane to keep the concentration equal. Now water or solvent molecule move as follows from region of to the region of. Okay, so from region of pure solvent it goes to the region of solution. From region of dilute solution it goes to the region of concentrated solution. From the region of high free energy of water molecules to low free energy of water molecule, and from high water potential to the lower water potential, and from higher diffusion pressure of water to the lower diffusion pressure of water. Okay, now we'll talk about the osmotic pressure, what it is. So Pfeffer, he was the scientist, he coined the term osmotic pressure. Osmotic pressure of a solution is a pressure which must be applied to it in order to prevent passage of solvent due to osmosis. Okay, osmosis, or you can say osmotic pressure of a solution is a pressure which must be applied to it in order to prevent the passage of solvent due to osmosis. Okay, and osmotic pressure is equivalent of maximum hydrostatic pressure which is produced in the solution when this solution is separated from its pure solvent by a semi-permeable membrane. So osmotic pressure, you can say that it can be defined as excessive hydrostatic pressure which must be applied to it in order to make its water potential equal to that of pure water. See herein, this is U-shaped beaker, and this is osmotic pressure which is applied to stop osmosis, and this is the semi-permeable membrane, this is the solution, and this is the pure solvent. So it is applied to stop the osmosis, or you can say that it can be defined as excessive hydrostatic pressure. See, this red arrow is showing excessive hydrostatic pressure which has applied to it in order to make its water potential equal to that of pure water. So osmotic pressure is equal to the pressure which is needed to prevent the passage of pure water into an aqueous solution through a semi-permeable membrane. In other words, it is that pressure which is needed to check the process of osmosis. Okay, I hope it is clear to you now. Now we'll talk about the water potential, students. It is the movement of water in plants that cannot be accurately explained in terms of difference in concentration or in other linear expression. Okay, the best way to express the spontaneous movement of water from one region to another is in terms of difference of free energy of water between two regions. Okay, free energy is what? It is a thermodynamic parameter that determines the direction in which physical and chemical change must occur. And the potential energy of water is called as water potential. For example, water is stored behind the dam. So it has water potential energy. The other source that provides energy to water is pressure. The increasing pressure, it increases the free energy, thereby increasing the water potential. See, this is the distilled water which has water potential 0. This is the plant cell that immediately after being put into a distilled water, see, this is what it is. So its water potential is in the negative form. And this is the plant cell after being in distilled water for some time. Then it applies the turgor pressure also. So this is +2 and this is -2. Therefore it is 0. Okay, I hope it is clear to you that water potential is the movement of a water in plant that cannot be accurately explained in terms of difference in concentration or in any other linear expression. Okay, now we'll talk about the osmotic or solute potential. It is a decrease in the chemical potential of a pure water due to the presence of solute particles. See, when you add solute particles in the pure water, then decrease in the chemical potential of a pure water takes place due to the presence of solute particles in it. And solute potential, it is colligative property of a solute and is dependent upon the number of solute particles and not upon the nature of solute. See, osmotic or solute potential, it does not depend on the number of the solute particles, but it depends on the nature of the solute particles. Solute particle, it reduces the free energy of water by diluting it. Thus, it increases the entropy. Entropy is a free energy, and thus it reduces vapor pressure, and thus it raises the free energy of water by diluting. By increasing entropy, by reducing vapor pressure, and thus it raises the boiling point and it thus lowers the freezing point. So this is the solute potential, is the tendency of water to move by osmosis. See, this is the solution, these small ones are the solutes, and this is the plant cell, and this is the vacuole of a plant cell, this is the mitochondria of a plant cell. And solute potential inside cell and in surrounding solution is same. No net movement of water takes place. And when cell is placed in pure water, its solute potential is low related to its surrounding, and water moves into cell via osmosis. Okay, I hope it is clear to you now. Now we'll talk about the pressure potential or hydrostatic potential, students. It is a pressure which develops in an osmotic system due to osmotic entry or exit of water from it. Positive pressure, it develops in the plant cell due to entry of water. Okay, so positive hydrostatic pressure is called as turgor pressure, and the loss of water produces a negative hydrostatic pressure or tension, and it develops in xylem due to loss of water in transpiration. So pressure potential is the tendency of water to move in response to pressure. Whenever you apply pressure, then water moves, or it has the tendency to move. and that is called as pressure potential. Turgor pressure is an important source of pressure on water in cells, and this is very important in transport of sap over long distances in plant. I repeat, it is very important in transport of sap over long distances in plants. See, this is plasma membrane, this is the cell wall of a plant cell, this is the Turgor pressure, which is cell is applying from inside of the cell, and thus it expands the volume of a cell, and thus it pushes membrane out. And this is the wall pressure that is being applied from outside on the cell wall, and a stiff cell wall it pushes back with the equal and opposite force. Okay, so this is very important in transport of sap over long distances in plants. Now, what's the significance of osmosis in plants, students? It is the phenomena of osmosis is important in absorption of water by plants, obviously, and cell to cell movement of water occurs throughout the plant body due to osmosis. The rigidity of plant organs such as shape and form of organism is maintained through osmosis. Leaf, they become turgid and it expands due to their osmotic pressure. Growing points of root, it remains turgid due to osmosis and are thus able to penetrate the soil particles. And resistance of plant to drought and frost is brought about by osmotic pressure of their cells. Movement of plant and plant part is also because of the osmosis. And one foremost and crucial importance of osmosis is opening and closing of stomata. It is affected by osmosis. Okay, now we'll talk about the plasmolysis, students. See, this is the normal plant cell. If a living plant cell is placed in a highly concentrated solution, that is hypertonic solution which has a higher concentration solution, then water comes out of a cell due to exoosmosis through a plasma membrane. See, this is the water dilute solution, and this is the concentrated solution, and this is the plant cell which is placed inside the concentrated solution. So cell becomes plasmolized. That means loss of water from the cell sap. It causes the shrinkage of the protoplast away from the cell wall in the form of round mass in the center. And the shrinkage of the protoplast of a living cell from its cell wall due to exoosmosis under the influence of a hypertonic solution is called as plasmolysis. I repeat, shrinkage of a protoplast of a living cell from its cell wall due to exoosmosis under influence of highly concentrated solution, that is hypertonic solution, it is called as plasmolysis, wherein protoplast pulls away from the cell wall. Okay, plasmolysis, it depends on the three type of solution. First is isotonic, hypotonic and hypertonic. Isotonic, it is a condition when external solution balances the osmotic pressure of a cytoplasm. That means see, this is controlled by this. Okay, so it is a condition when external solution it balances the osmotic pressure of a cytoplasm. Then comes hypotonic. See, this is hypotonic. In this case, external solution is dilute as compared to cytoplasm. So, water moves in. See, this yellow arrows are showing that water is moving inside and cell becomes turgid. So, in hypotonic solution, cell becomes turgid. I repeat, in hypotonic solution, if you keep plant cell, then cell becomes turgid. Then comes hypertonic solution, in which external solution is more concentrated. So, cell sap it moves out. See, this green arrow is showing that water is moving out and cell has become plasmolized. Okay, so these are the three types of solution: isotonic, hypotonic and hypertonic. So the stage of plasmolysis when protoplasm just begins to contract away from the cell wall is called as incipient plasmolysis. See, this is incipient plasmolysis when protoplasm just begins to contract away from the cell wall. The stage when cell wall has reached its limit of contraction and protoplast has detached from the cell wall and it attains a spherical shape, then it is called as evident plasmolysis. When protoplast detaches itself from the cell wall and thus it attains a spherical shape, then that is called as evident plasmolysis. In a plasmolized cell, the space between contract protoplast and the cell wall, it remains filled with the external solution. And if a cell with incipient plasmolysis is placed in hypertonic solution, it will show more plasmolysis. I repeat, if a cell with incipient plasmolysis, I just taught you, is placed in the hypertonic solution, it will show more plasmolysis. And if a plasmolyzed cell is placed in pure water or hypertonic solution, then endoosmosis takes place. That means reversal takes place. Water is taken up inside the cell. Okay, that is called as endoosmosis. So reversal takes place if you keep plasmolyzed cell in pure water or hypertonic water. Now we'll talk about the deplasmolysis, students, wherein the protoplast, it attains its original shape and cell, it regains its original size. The reversal of plasmolysis is deplasmolysis, wherein protoplast, it attains its original shape and cell, it regains its original size. And the swelling up of a plasmolyzed protoplast due to endoosmosis under the influence of hypotonic solution. See students, you can memorize it like hypo endo, that means hypotonic solution, endoosmosis will take place. So hypo and endo, this way you can memorize it. Okay, and deplasmolysis is possible only immediately after plasmolysis, otherwise cell protoplast becomes permanently damaged. Leaf of Tendentia is used for demonstration of plasmolysis in laboratory. Okay, this is usually it is seen that a leaf of Tendentia is used for demonstration of plasmolysis in laboratory, and the value of Turgor pressure, that is TP, becomes 0 at the time of limiting plasmolysis, and it goes below 0 during incipient and evident plasmolysis. Okay, this is showing that when you keep plant cell in a hypotonic solution, that water moves inside it, and the value of the Turgor pressure becomes 0 at the time of limiting plasmolysis. Now, what are the significance of plasmolysis, students? It proves that the permeability of cell wall and the semi-permeable membrane of a protoplasm, and the osmotic pressure of a cell can be measured by plasmolysis. So that's the crucial significance of plasmolysis, that osmotic pressure of a cell can be measured by plasmolysis. And osmotic pressure of a cell is roughly equal to the osmotic pressure of a solution that causes incipient plasmolysis in the cell. For example, salting of pickle, meat, fishes, and addition of sugar to jam, jellies, cut fruit, etc. it prevent their decay by microbes, as the latter get killed due to plasmolysis or due to high concentration of sugar or salt. By salting, weeds can be killed from tennis courts, and the growth of a plant can be prevented in the cracks of walls. Okay, so that's the significance of plasmolysis, and the plasmolysis is helpful in determining whether particular cell is living or dead. I repeat, plasmolysis, it is helpful in determining whether a particular cell is living or dead, as plasmolysis does not occur in dead or non-living cells. Okay, now last topic is guttation. Guttation is the exudation of drop of xylem sap on these tip or edges of the leaf of some vascular plants such as grasses. See, this is showing guttation. Guttation should not be confused with dew, which condenses from the atmosphere onto the plant surface. So transpiration, it usually does not occur in night due to the closure of stomata. See, when stomata is open, then transpiration will take place, but when the level of moisture is high in soil, the water enters in the plant roots. Okay, and this water is accumulated in plant, resulting in increase in root pressure, and this root pressure, it forces some water to exude out from the edges of these leaves or edges of the leaf tip. The process of guttation is noticeable in case of suppression of transpiration and increase of relative humidity during night. That means guttation only takes place when high moisture is there, and when you suppress the transpiration and increase the relative humidity during night. So guttation is noticeable in these two cases. Okay, So this was all about it. In my next section of the presentation, we'll be discussing about the vascular bundles. So till then, stay tuned and keep watching Edupedia word videos. Thank you.